Why don't we go ahead and get started with my stuff, and we'll let people just continue to join as they come in. Thank you everyone for joining us. My name is Cinnamon Moffett, and I'm the Research Program Manager at Oregon State University at Hatfield Marine Science Center in Newport, Oregon. And you're here today, hopefully, for the HMSC Research Seminar. I'm going to be your host today, and I'm just going to talk a little bit about some logistics and some announcements, and then we'll get started. So just wanted to let everybody know that your mics, camera, and screen shares have been turned off. It's really helpful if you can keep them turned off for us during the course of this event, just to help folks that might have low bandwidth really focus on our speaker today. So I appreciate that. You are encouraged to put any of your questions into the chat box. You can find that either at the bottom or the top of your screen, depending on what platform you're using. Feel free to put in those questions at any time and we will get to them either during the talk or after. I also wanted to let everybody know that we are recording today's event. So if you happen to miss something or you want to share it with your colleague, it should go up on the HMSC past seminar page in a couple of days. And I am putting that link into the chat box right now, so you have that. Couple of quick announcements before we get started. Next week's seminar, same place, same time, just on April 8th. We have James Watson coming to speak to us about the social environmental analysis. Oh, sorry, he's from the Social Environmental Analysis Lab as CEOS at Oregon State University. And he's going to be talking about the living with non-stationary climate change, risk management for social ecological resilience. And we've had James speak with us before, and it's always an interesting conversation. So I hope you're able to join us again on April 8th. A couple little plugs, just want to talk about Virtual Marine Science Day next weekend. If you're interested in that event, it is running from 10 to 2. And if you want information on Marine Science Day or any of our events, you can Google or use whatever platform you use to put in HMSC, scroll to the bottom of our homepage, and there's a calendar of events there with all your login details that you might need. I also was asked by Yi-Chung to let everybody know, if you're a student online here, that there's going to be an information sharing meeting next week, actually a couple of them, about scholarship opportunities. So if you're interested in applying for any of the scholarships, check out HMSC's academic page on the web and look for scholarships, and information and login information for those informational meetings will be there. But for today, we have Ryan Mueller here to talk to us about phytoplankton and the marine microbial loop. But first, just a little bit of background about Ryan. Ryan received his PhD from the University of California, San Diego, working in Professor Bartlett's lab. After graduating, he completed a postdoc with Professor Jill Banfield at the University of California in Berkeley. He is now an associate professor at the Department of Microbiology at OSU. His lab investigates microbial interactions with different ecosystems that drive biochemical cycles, influence microbial community assemblages, and affect the host microbiome relationships. And he is working with developing methods for studying those interactions, both in situ and in the lab. And I have a feeling he's going to talk to us a little bit today about how some of those things have been applied. So Ryan, the floor is yours. I'm going to turn off my mic and camera and hand it off to you.
Great. Thank you so much for the invitation. Thank you everybody for joining me. Now I just get to stare at a bunch of black boxes, but good thing I'm used to it after about a year of this. So let's go ahead and share my screen and we'll get started with the talk. So here we go. All right, so we're going to be talking about partitioning of phytoplankton organic matter by the marine microbial loop. And this is a project that was started pretty much when I first came to OSU by one of my first graduate students, or actually my very first graduate student, Sam Bryson, and then continued by my second graduate student, Brandon Keefe. And this process, or this project, really was focused on developing methods in order to track activity of different microbial populations in marine systems. So as Cinnamon pointed out, I came from UCSD, Scripps Institution of Oceanography. So I have an actual PhD in marine biology, but I rarely go out to sea. And for this particular project, we basically waded in the Yaquina estuary and pulled out some water in order to look at the microbial communities found in the estuary here and how they process organic matter. And so we're going to be focusing more or less conceptually, at least, on this area that's shown in these pictures. So the phycosphere, so the area around phytoplankton that are found in the marine environment. These beautiful pictures were taken from Marine Particles and Phycospheres Conference 2019, basically showing really these very small scale environments. So around single cells of either active or dying phytoplankton, cyanobacteria, and all of the bacteria. So this picture in particular really is a microscope image showing the intense activity of bacteria surrounding this one particular diatom cell. So we're going to get into that and think about the activity at this very small scale and what's happening with the organic matter that's being produced by the phytoplankton. Okay, so as Cinnamon said, if you have any questions as we go through this, please add them into the chat. I'll be trying to look out for them. Cinnamon can prompt me as we go too. So let's get started here and start setting the scene by looking at this beautiful picture of what I believe is a cyanobacteria bloom in the Baltic Sea. And I wanted to start here by thinking about blooms in general. Phytoplankton blooms caused by eukaryotic phytoplankton and cyanobacteria, prokaryotic cyanobacteria, and thinking about how they alter the system. We know phytoplankton blooms will occur annually during spring and summer seasons, regularly in different parts of the globe, usually along the coastlines. And in doing so, these phytoplankton blooms will rise up and accumulate relatively rapidly within maybe days or weeks. If the conditions are right, the right physical, chemical conditions all lead to extreme phytoplankton growth, leading to these blooms, which leads to massive amounts of activity, both photosynthetic activity, metabolic, catabolic, anabolic activity, leading to growth of these phytoplankton and continued on and on of use of resources, production of organic matter, ultimately until that bloom starts to decline for whatever reasons. The physical, chemical environment changes, the resources get used up, the phytoplankton start to die. And obviously something happens to all the organic matter that's being produced. And so more or less, that's the point of the talk here. What's happening to that organic matter that's being produced? So we have this major change in the ecology of these systems as these phytoplankton blooms occur and decline. And so we wanted to explore that a little bit. And so the relevance here, let's try to think about the processes of what's happening. We think about the marine carbon pump. With phytoplankton accounting for 0.2% of the Earth's photosynthetic biomass, but almost 45% of the total primary production that's occurring annually. So this is always an amazing statistic. And specifically to a microbiologist as myself, because you really see the power of microbes here. We have these phytoplankton, these single-celled organisms you can barely see with naked eye, often can't. Many of them are truly microscopic. And yet, when we integrate the total activity across the global scale, we see that these tiny microbes are producing about 50% of the annual primary production on this planet, which is just truly incredible. And importantly, these phytoplankton are going to take CO2 from the atmosphere, dissolve CO2 in the water, and then turn it into fixed organic matter, right? As a major step in global carbon cycles, the fixation of inorganic carbon into organic carbon. And when we think about the carbon pump, we're typically simplifying this into thinking about a dissolved pool of organic matter that's being fixed by these phytoplankton. So all the small molecules, organic molecules that these phytoplankton are producing, simple sugars, amino acids, etc. And then we have our particulate organic matter. So this would be the non-dissolved solid pieces of material, biomass that these phytoplankton are producing as part of their growth and metabolism. So 6 to 33% of that fixed carbon that's being fixed annually by these phytoplankton is thought to be released as dissolved organic matter. And the dissolved component is really important for what I am interested in studying, namely the heterotrophic microbes in the microbial loop, because they are the ones that are primarily using that dissolved organic matter. But when we think about the marine carbon pump, we have these two pools of carbon. And then the question is, what happens to that carbon? So after it's produced, where does it go? Well, in another simplistic idea, we have the idea that some of this carbon is going to be shunted into organisms that feed directly on the phytoplankton through grazing. So we have larger organisms that are eating the phytoplankton, or through organisms like the bacteria that are using that dissolved organic matter. Ultimately, those organisms, those heterotrophs, those consumers, are going to take that carbon in and then make a decision of whether to use that carbon for respiration, energy production, or alternatively for biomass, so anabolic processes. And ultimately, this can greatly influence how much carbon is being released back into the atmosphere through mineralization processes, or being retained in aquatic food webs through the production of biomass and the transfer of that carbon through the different trophic levels. If that carbon isn't being used by the consumers, we tend to think of it as potentially aggregating. And this is particularly true when we have a phytoplankton bloom that occurs and then declines, and all of those phytoplankton die. Ultimately, that detrital material can aggregate, form marine snow, and then sink towards the bottom of the ocean. Obviously, it can be colonized by microbes and start to be respired and mineralized through that sinking process. But a significant portion of that organic matter can sink into the marine sediments where there's very low microbial activity and overall activity, meaning that effectively that carbon, that organic matter, is buried and sequestered in the marine sediments for very long periods of time. So this is a very important step, because we've gone from taking the CO2 in the atmosphere, putting it into a biological active form, organic matter, and then that organic matter is either being actively cycled in the surface ocean, throughout the water column, or it's being buried into the marine sediments. And so obviously all of this transitioning of the carbon can greatly influence global carbon cycles. So let's talk a little bit about that first step of use of this carbon by the marine microbial loop. So the consumption of organic matter that's produced by phytoplankton. So the microbial loop, we're defining that as the heterotrophic bacteria, archaea, flagellates, other microzooplankton that are basically eating the organic carbon from phytoplankton. The phytoplankton primary producers, here we're talking about the primary, secondary, tertiary consumers of that carbon. And we're talking about the microbes that are part of this system, part of this food web. Interesting note here, heterotrophic microbes are thought to consume between 10 and 50% of the primary production of phytoplankton. So we said that phytoplankton on a global scale are producing about 50% of the world's carbon, fixing that annually. Here we have about 10 to 50% of that being used directly by heterotrophic microbes. So ultimately 5 to 25% of the total carbon that's being fixed annually is flowing through these tiny heterotrophic microbes that are out in the world's oceans. So these organisms are a really important part of global carbon cycles and dictate the flow of carbon. An important kind of directional consequence of this is that we can have that carbon either be retained in the system, as I indicated on the previous slide, through trophic transfer events, or we can have that carbon released back into the atmosphere or the water column as inorganic carbon, CO2, DIC, and ultimately lost into the atmosphere. And so this is a reproduced figure from one of the seminal papers on the microbial loop, the paper that first named this as the microbial loop, showing exactly what's going on here from kind of a mechanistic model point of view, where we have cyanobacteria, phytoplankton down here at the bottom. They're the primary producers in the system. They're producing that dissolved organic matter, the particulate organic matter, and the bacteria specifically are using, they're pretty much the sole consumers of the dissolved organic matter. They're the ones that secrete enzymes into the environment. They have transporters on the outside of their cell that allow them to take up these dissolved organic matter materials. Those bacteria can obviously be consumed by larger predators like flagellates, Those can be consumed by ciliates, zooplankton, and then on up to fish, let's say. So here we're basically seeing how that organic matter can transition from that primary point of fixation into consumption and secondary and tertiary consumption, ultimately leading to carbon flow through the different biomass of the different trophic levels of the system. But the one side that we have to keep in mind here is that a significant portion of that carbon is going to be remineralized at each one of these steps. And when we think about bacteria, that's going to be dependent on which bacteria are actually using that dissolved organic matter, meaning that bacteria have different growth efficiencies. Some use more carbon for respiration and energy production, and others use more carbon for biomass. And so the bacteria present and the ones that are using that carbon can dictate how much carbon is lost back into the atmosphere versus how much carbon is retained in the biomass of the system. So here we have our global carbon cycle, very nice image of this. And I'm sure all of you are familiar with this, so I'm not going to go into too many details, but I want to point out a couple key things before we move on. So first thing is pretty obvious probably to everyone here. We're talking about global carbon cycles. So we're talking about immense scales, gigatons of carbon per year that are flowing in and out of the different pools of carbon along the planet. And so, this is a major process in terms of what's going on here. The other thing I want to mention, again, probably pretty obvious at this point, especially given where we are at in the planet these days, is whenever you take a whole bunch of carbon from one pool and move it to another pool, like say, take a whole bunch of carbon in the fossil pool and then release it into the atmosphere through combustion, that's going to change the system. It's going to dramatically change the system in many ways and lead to some unintended or unexpected consequences. And obviously, we're dealing with that now. The last thing I want to point out is who are the organisms that are dictating the flows of carbon? Well, it's obviously a lot of the organisms that we're used to thinking about, these large organisms that are on the planet, but the microbes are really important for this process. As I pointed out, phytoplankton are dictating the flow of carbon from the atmosphere into fixed biomass at huge rates. And the microbes are really important. Marine microbes specifically are really important in respiring that carbon back into the atmosphere. If we think about the terrestrial side, same sort of thing, except now we have plants that are fixing that carbon, but the soil microbes are the ones that are important for releasing that carbon, that stored soil organic matter, back into the atmosphere. So microbes are really playing a huge role in these global carbon cycles. So where are we at in terms of thinking about global carbon cycles? Well, we're really good at sending up satellites into space and monitoring through remote sensing primary production. And so this is a recent paper that came out basically showing primary production patterns in the global oceans, in this case, the North and South Atlantic over time. And by doing this type of research, we can begin to get a better understanding about global variations in the processes that drive carbon cycles. So as you can kind of clearly see here in this image, we see that the primary production is changing throughout the year. We expect this, right? We just said blooms are going to occur annually at defined times of the year. And not only does it change seasonally, temporally, but we're also going to see spatial changes. We're going to see changes that are related to different Earth processes like El Nino or different oscillation events that all come to effect when and where there's greater or lesser primary production in the global oceans. Ultimately, these long-term observations can help us build models about global carbon cycles and ultimately help to tell us something about climate, right? And how climate might be impacted by the changes in primary production or changes in overall carbon cycling in the system. And so one thing I like to think about here is not just this large scale processes, these global scale processes that can be tracked with satellites, remote sensing, but how to connect these large scale processes down to the micro scale. and thinking about how individual microbial metabolism, individual cells, individual phytoplankton and bacteria, and how the interactions between those can really drive the cycling of carbon at global scales. And to emphasize this scale, this tiny scale, I love to show this movie from an old colleague of mine, Steve Smirga, when he was doing his postdoc from a few years ago, which is basically showing what we're looking at here is a microscope image. We're going to see a movie of a diatom cell that's fixed in position on this microscope slide. And all the little specks that you see around that diatom right now, those are going to be bacteria. The bacteria are about 1 micron, 2 microns in scale, and the diatom's probably about 20 to 30 microns in scale. And what we're going to see is as this movie progresses, this diatom is being hit by a laser. That laser is exciting the fluid in that diatom, ultimately heating it up, and that diatom is going to burst. And what I want you to see is the difference in the behavior of the bacteria around that diatom as we go from a state of non-burst cell to effectively killed cell, where now the organic matter that was inside of the cell is leaking out into the environment. And basically, we're seeing this huge swarm of activity almost immediately, within seconds. I think this is sped up maybe 1 or 2 times, but it's not sped up that much. So within less than a minute, we have this huge swarm of bacteria that are sensing the chemical gradients that are emitted from that lysed diatom cell. So you can just see how dramatic the phycosphere is impacted by the chemical gradients that these diatoms, or phytoplankton more generally, are producing as they're growing and as they might die. If we think about an entire phytoplankton bloom, you go back to that first slide and think about that large bloom. If all of those cells die at the same time, what happens to the microbial activity as those cells start to release their dissolved organic matter? So this is where we're interested in, and we're interested in asking, what are these microbes doing in these systems? But of course, there's challenges to studying these microbes. The main challenge is really to link function to the distinct types of bacteria. In that previous movie, we're not telling which bacterium is distinct from each other bacterium. They all look like bacteria. And so how do we tell the individual bacteria apart from each other? And then also tell which bacteria are doing different things in the environment? This is not a simple thing to answer. And we've come a long way, though, by using omics techniques, genomics, proteomics, transcriptomics. Basically, all these techniques is taking all of the RNA, all of the protein or DNA, and sequencing the heck out of it in order to say which genes are there, which proteins are there, and which transcripts, RNA transcripts are there. And this can tell us something about potential metabolism. We're not getting direct functional evidence of any sort of thing going on in these samples, but by seeing that a gene is present, we have the idea and we can hypothesize about which functions are being performed by those bacteria. But we're still missing these direct measurements of activity of individual populations in these systems. There are methods out there that can do this, and these are typically based on isotope tracer methods in order to measure function directly in systems. And the basic idea here is it's a simple incubation experiment where you take a particular compound that you're interested in looking at its metabolism by microbes, and you label it with an isotope. So this can be a radioactive isotope or a stable isotope. If we're talking about carbon, we're talking about C14 or C13. Ultimately, you add the substrate, incubate for a certain amount of time, and then you can look at activity of the microbes. So basically their uptake or utilization of that substrate at the single cell level with methods like MAR-FISH, or individual populations where you use DNA stable isotope probing, or you can look at bulk methods to look at the entire community utilization of that substrate. So basically we're adding like radioactive glucose, let's say, and then we're looking at the release of radioactive carbon dioxide from that incubation. But all of those methods have some drawbacks. So we were determined to try to advance the methodology a bit in order to understand this process at a more resolved level. And this was really started with my first graduate student, Sam Bryson, and Chongo Pan, who was a collaborator at Oak Ridge at the time, who's now at University of Oklahoma, and Bob Pettit at Oak Ridge, in order to basically take what would be considered traditional metaproteomic techniques, so mass spectrometry-based proteomics, and combine it with these stable isotope probing methods. And the basic idea, just same experimental design, we're adding a labeled substrate, incubating the microbes, but now instead of just kind of measuring the analyte in terms of how much CO2 is released, radioactive CO2, we're actually looking for incorporation into the proteins that are being produced after substrate addition. And so basically we collect all the cells, extract proteins from those cells, digest those proteins in the peptides, and then run all those peptides on a mass spec in order to determine which peptides are present. This is basically the traditional part of metaproteomics. That's what you would do in a proteomic experiment. The other side of this is where we take those same cells that we've collected and extract DNA. And this is to basically do DNA sequencing and metagenomics so that we can develop a peptide database in order to say, what are all the potential proteins that can be expressed in our community? Because now we have spectra that we have to match to known peptides. And so basically at this step, we take our peptide database of potential proteins and match it against the detected spectra that represent the real peptides in our sample. And because this is a stable isotope probing approach, we use a sophisticated algorithm in order to basically say which peptides are present and how much are they labeled. And this is a very computational intensive approach. And so we typically have to use supercomputers to do this. But a couple key points. The mass spectrometry is very high sensitivity and has very high resolving power. So basically, this allows us to really take and advance this SIP labeling approach methodology in order to detect activity in our samples. So we can use very short incubation times of 12 to 16 hours instead of multiple days. This allows us to focus on the primary consumers of the resource and not cross-feeding events that happen after extended periods of incubation. And it allows us to use a lot less substrate than would be used in other approaches like DNA SIP. So we still have to add about 1 micromolar to our samples. So it's not as sensitive as some radioactive labeled methods, but 1 micromolar is certainly a lot less than what would be added of 100 or 10 micromolar for DNA SIP approaches. So what can this proteomic SIP data tell us? Well, essentially we're collecting mass spectra, right? Ultimately, those mass spectra are going to give us a peptide sequence. So I have a generic peptide sequence represented here. We can take that sequence and match it to known proteins. By doing that, we can say which enzyme encodes that particular peptide. And if we know which enzyme is, we can basically say something about what is the function that is carried out by that protein that is detected in our sample. On the other side of it, because we have sequence data, we can also say which organism expresses that protein. We have our metagenomes, we can go back to the genes, translate those genes into amino acid sequences and match that up and basically use that information to say which organism it came from, because there's phylogenetic information in this peptide sequence as well as functional information. The other thing that's special with proteomic SIP is the SIP part. We can quantify how much 13C label, how many atoms in all of the carbon atoms are 13C versus the 12C. And we can do this across the entire range of detectable amounts of 13C from 2%, which is about natural abundance of 13C in the environment, all the way up to 100% labeling, which would be very abnormal for natural conditions, but would be within the realm of possibility for experimental conditions where we're adding 100% labeled substrate. So ultimately, this can tell us what is the proportion of 13C in that peptide. But of course, we're not just focusing on one single peptide here. We're focusing on thousands of potential peptides, tens of thousands, from hundreds of thousands of different spectra. So ultimately, we can summarize peptides into the protein level and then summarize peptides into functional groups. So similar sets of proteins, homologs of each other, that all do the same thing. Gather all the ribosomal proteins together, and we classify those as one group. Or we can summarize the peptides at the protein level and group them into the organism groups that we're interested in looking at. And so basically, we can take these groups of proteins that we detect in our samples and say something about the overall labeling within the total group. And so that's where we've come up with a couple metrics to measure labeling efficiency in these samples. One metric we call label frequency, LF, and that basically is just telling us, we ask, is a protein labeled or is it not? And then of the group of proteins, what proportion are labeled above natural, above 2%, or are not labeled? And this tells us something about the overall activity of the cells in our sample. The other metric, average enrichment, is saying when we only consider the label proteins of our group, organism or functional group, what is the average enrichment value? So now we're looking across this sliding scale and we say, are all these peptides above 70% enriched or below 20% enriched? And this really is able to tell us something about the overall activity and the substrate specialization of the organisms in our samples. So the last kind of intro background that I'll get into, resource partitioning. This is how we essentially use this methodology. We think about how organisms are using substrates and competing for substrates in natural environments. And so basically the schematic I have here is we add our label substrate. That substrate can be partitioned and used at different rates by different organisms in our system. So basically we have some winners and some losers, meaning that this taxon at the top is it really likes glucose, so it gets heavily labeled. And this taxon at the bottom doesn't really mind glucose, doesn't use it, and so there's barely any label in that particular group. And when we look at the protein level, we can see differential labeling across the proteins. So ultimately, this is telling us something about the response of that organism to that substrate. Because remember, we're only going to detect label in proteins that are newly synthesized. That's how the label will get into those proteins. So basically, any proteins that we see that are labeled are being expressed directly after the substrate has been added to our incubations. So we can say something about the response, the metabolic response, the enzymatic response of these organisms to substrate additions. Okay, so our particular experiment here that I want to discuss in about the last 30 minutes or so, 20 minutes, our research question was basically, do different bacteria of the microbial loop specialize on distinct phytoplankton dissolved organic matter pools? Focusing on this first step, we have carbon fixation. What's that primary consumption step? Who is getting that organic matter, and is there different specialization depending on where the organic matter is coming from? And so we used two model phytoplankton that we grew in the lab, the Lasiosira pseudonana, a diatom, really important in global carbon cycles, and then Synechococcus, a cyanobacteria. So we have a eukaryote and a prokaryote, very different organisms, very different physiology, but they both cause blooms. They're both important in primary production on global scales. In addition to using these two model phytoplankton, we wanted to use two different substrate pools. So basically we took the exudate fraction, so everything that's released by the phytoplankton as they're growing in our culture conditions, and compared that to uptake of the lysate fraction, which was meant to emulate basically when these blooms decline, all the cells die, and all of that internal guts of the phytoplankton gets released out into the environment. And so basically we have the actively released organic matter, dissolved organic matter, versus the organic matter that's released after the death and the lysis of these phytoplankton cells. So we have those four treatment conditions that we were looking at. Basic experimental design, we grow up the diatom, the cyanobacteria, under labeling conditions, so 13C bicarbonate in the cultures, and then collect the exudate, lysate. And then ultimately, we're incubating with water that was collected from Yaquina estuary, as I indicated. So basically, natural microbial communities incubated with these label substrate pools, extract proteins, extract DNAs, do all the proteomics, the genomics, all that fun stuff, and then analyze the data for what ultimately turned out to be several years. So these are the results. Basically, what we're looking at here on the left-hand side of the slide is the taxonomic composition of our samples, taxonomic structure of our samples. So we have our control T0, so before any substrate addition, that's how we're labeling this, and the different organisms that were found and the different relative abundances of the proteins, because this is all proteomic data being represented here. T1 is no substrate addition plus incubation. So 15 hours of incubation, what happens to the community in the community proteomes? And then here are all of our substrate addition plus incubation. So again, 15 hours after adding the exudate from cyanobacteria, the lysate from cyanobacteria, and the exudate and lysate from diatoms. And when we can see some shifts occurring here, you can see some shifts in the community structure. And that's summarized here on the PCA plot on the right, where we see that the T0 samples, so our three replicates of the pre-incubation community, is off by itself from all of the other samples. And then our other set of samples, the T1, so post-incubation, but no substrate addition, we also have a distinction from all of the other samples where all of the treatment samples are clustered together. So basically, we see an incubation effect and we see a substrate effect. But when we're looking at all of the proteomes, so both labeled and unlabeled proteomes, basically, all of the substrate additions seem to create the same effect. There's no significant difference between the different substrate treatments when we're considering all of the proteins. And that's reinforced here when we just look at the unlabeled metaproteomes, so the 12C detected proteins in our samples. There's no significant difference between the treatments. Very different, however, between what we see when we focus exclusively on the 13C metaproteomes. So remember, these are the proteins that are being produced de novo, so being produced as these organisms are growing in response to the substrate additions. So the proteins that are produced as a result of that substrate addition, we see that they're very distinct from each other based on the different treatments that we subjected them to. And the last thing I'll go back to are these metrics of enrichment value, average enrichment, and label frequency. And we're just measuring these, which are basically just telling us how much label was incorporated into the biomass across the different treatments. So we have our lysate down to cyanobacteria exudate diatom on the left. And basically we see a decreasing effect where from the diatom lysate, we have the greatest assimilation of the carbon substrates, so the diatom lysate here. And over on the right-hand side, the cyanobacteria exudate is the least assimilated by all of the bacteria in our samples. So we do see this treatment effect borne out at the community level when we focus on the labeled proteomes of these samples. And that's reinforced again here, where we're looking at the proteomes, again, the labeled proteomes, and looking at the taxonomic structure of those proteomes. So because we're not focusing on the unlabeled, the T0 and T1 samples aren't in this graph because there's no labeling there. There's no labeled substrate addition. But you see very big changes across these treatments in the community composition. So this is reflective of the different organisms that are producing proteins directly as a response to those different substrate additions. And we'll go into some of the details in the next couple of slides. So the main groups that we're going to focus on are Bacteroidetes, alpha proteobacteria, and gamma proteobacteria. And the taxa that we detected in our samples are very common across all the different marine samples that we've collected along the Oregon coast. And each of these typically constitutes about 20 to 30% of a given sample. So these are always the dominant taxa in our samples, no matter if they're exposed to substrate incubations or not. This is the natural condition. So let's focus a little bit on the alpha proteobacteria. And what we're looking at here on the left-hand side is a histogram showing how labeled the proteins from the alpha proteobacteria were in the different treatments. And the main takeaway here is that across all of the treatments, the alpha proteobacteria are always pretty well labeled. So our average enrichment for the labeled proteins is about 60 to 70 across all of the different treatments. But we do see a treatment effect where the organisms in the diatom lysate are slightly less overall labeled than the cyanoexidate relative to all the other organisms in our sample. So basically, the alpha proteobacteria seem to do best in the cyanoexidate treatments, and they do worse in the diatom lysate treatments. This is very different than the Bacteroidetes patterns, where, again, we see very high labeling, but not in all of the treatments. Specifically, the cyanobacteria exudate treatment had relatively low labeling relative to all of the other treatments. And one thing I want to point out here, specifically with the Bacteroidetes, we have a significant portion of proteins that were detected that were 100% labeled in the diatom, exudate, lysate, and the cyanobacteria lysate samples. These Bacteroidetes, after 15 hours, have basically turned over their entire proteins, single proteins, and exclusively use the substrates that we've added to our incubations in order to synthesize new proteins during these incubations. So this is really kind of striking to me, the time scales involved in the synthesis and utilization of these substrates. Moving on to gamma proteobacteria, we see something more similar to the Bacteroidetes. So we again see pretty high labeling in diatom lysate, exudate, and cyanobacteria lysate, but in the cyanoexudate, we see relatively low average enrichment values. So basically what this is telling us is the gamma proteobacteria, the Bacteroidetes, when they're using the cyanobacteria exudate, they're not solely using that carbon in order to synthesize new proteins. Only about 25% of the carbon atoms that are going into new protein synthesis are coming from that cyanobacteria exudate. This is different than the alpha proteobacteria, where 60 to 70% of the carbon atoms are exclusively coming from that cyanobacteria exudate. So basically, we're seeing some differences in the specialization on these different substrate pools by these different taxa. I also mentioned, in addition to the taxonomic information that we're getting from these samples, we can also say something about the functions encoded in those proteins. So these graphs, these ternary plots can be a bit confusing, so I'll try to take my time and walk through them real quick. But basically, we're grouping our proteins not just into taxonomic groups, but into functional groups. And the functional groupings are represented by the circles, and the taxonomic groupings are represented by the sides of these triangular ternary plots. And the way to view this plot is basically to look at where these circles are in relation to the corners of the triangle. If we see circles, so say category 9, which includes transcription and translation factor proteins, that circle, the size of the circle is relative to how many proteins were detected. But because that circle is in the center of this plot, basically it's telling me that that set of proteins, the transcription translation factors, was produced equally by alpha proteobacteria, Bacteroidetes, and gamma proteobacteria. They're all contributing equally to the production of transcription and translation factors. That's very different than, say, category 10, trap transport proteins, which is up in the corner, and specifically in the corner at the 100% end of the scale for alpha proteobacteria. So basically what this is telling me is the alpha proteobacteria are pretty much the only taxa that are producing trap transporters in our samples. The main thing I want to focus on these two graphs, so T0 versus T1, is that there's not a lot of change. So there's some community change, some community composition change that we see after incubation. But in terms of the functions that are being encoded in these proteins, it pretty much stays the same. The things that are in the center stay in the center. The things at the edges stay at the edges. So basically, the organisms probably are growing, some are dying, but they're basically producing the same proteins throughout that incubation period. This is very different than what we see for each of the treatments. And I'm just going to highlight the two extremes of these treatments, namely the cyanoexidate, where basically all of these shared proteins in the pre-incubation sample move up into the corner. And all this is essentially telling us is that in the cyanoexidate treatment, all of the proteins that we're essentially detecting that are labeled are from the alpha proteobacteria. This includes all of the growth and information processing proteins like transcription, translation factors. We see 9 moves from the center up towards this end of the corner of the graph. They're all being produced by alpha proteobacteria. The alpha proteobacteria are the active ones in the cyanobacteria exudate treatment. This is different than the diatom lysate treatment, where we start to see those growth and information processing proteins move down that axis along the Bacteroidetes side, indicating that the Bacteroidetes are becoming more and more active. And also the transporters that the Bacteroidetes produce, namely TUN-B dependent transporters, are being produced heavily in that particular treatment, the diatom lysate. So ultimately, this is telling us something about the functional responses and the growth responses of these taxa to the different treatments. We wanted to look at this a little bit more in detail, think about the growth responses and the physiology of these taxa under these different treatment conditions. And we wanted to ask basically, can we use these assimilation metrics in order to say something about growth rates of taxa in these systems? Because ultimately, what are we tracking here? We're tracking the new production of biomass, which is essentially growth. Enough new production of biomass accumulates, and you're going to have cell division of these cells in the natural environment. So we wanted to ask, basically, can we take these assimilation metrics and estimate growth? And in order to do that, we plotted out our assimilation values, basically our average enrichment multiplied by the label frequency as a general assimilation metric for each of the treatments in each of the 3 taxa that we've been focusing on, and then compared that against an independent metric of growth rate estimation made by using the metagenomic data that we had collected. So these are 2 very independent methods of estimating growth rate. And what we saw is that these 2 estimates of growth rate were very highly and significantly correlated with each other, basically indicating to us that we can use our assimilation metrics to say something about the active growth of bacteria in these systems, at the individual taxonomic level. We want to also ask something about, because the fact that we're seeing apparent resource specialization in our samples, do we see that the quality of the resource is reflected in the biomass of the bacteria that are using and specializing on that resource? And in fact, we see this. We see that these resource preferences are reflected in the carbon and nitrogen ratios of the bacteria that are using the different substrates in our samples. So let me break this down a little bit. Basically, we have our 2 substrate pools, and diatom are known to produce a substrate pool that has high CN values. So a lot of carbon, relatively low nitrogen. This is different than cyanobacteria organic matter, which is much lower, about 50% of diatom in terms of its carbon nitrogen ratio. So a lot more nitrogen relative to carbon. And so what we wanted to do is ask, if we have bacteria that are Bacteroidetes that are specializing on diatom organic matter, does that mean that their nucleotides and amino acids have the same carbon nitrogen ratio? So a high carbon nitrogen ratio relative to a lower one. And that's actually what we see. So basically we plotted out here the protein carbon nitrogen ratio and the diatom GC content. And when you have low GC, that's when you have a lot of A's and T's, which are more carbon rich. So basically, low GC means high CN. So basically, the Bacteroidetes, which are using the diatom, high CN material, are producing biomass that is reflective of that. The Bacteroidetes have high CN nucleotides and amino acids. And on the other side of the spectrum, the alpha proteobacteria, which we see uses the cyanobacteria exudate really well, that low CN ratio cyanobacteria exudate, they have a much lower ratio of CN in their amino acids and in their nucleotides that they're being produced. Last thing I want to wrap up with is the implications of all this and thinking about assimilation preferences on carbon cycling as a whole, thinking about phytoplankton blooms. So one way to do this is think about rates. We measured assimilation over time. Can we convert our assimilation metrics into some understanding or estimation of overall rates of carbon use and uptake in this system? And so that's what we did. And very clearly, we see big differences in the total assimilation of carbon from our diatom lysate, where we have well over 4,500 nanograms of carbon estimated being assimilated per liter per day in our samples, versus the cyanobacteria exudate, which is down around 1,500 nanograms, so almost a third as much carbon being assimilated and used. And importantly, it's not just driven by an overall community change, but specifically, we have the Bacteroidetes, which are really driving this dramatic increase in use of the diatom lysate materials in these samples. And so now we're seeing how an understanding of the individual population specialization and the resource partitioning can influence these overall processes and the overall rates and use of organic matter in these systems. So let's conclude. Let's wrap things up. If we're thinking about the marine system, a lot of us are concerned about marine systems and thinking about them from the perspective of the influence of anthropogenic stressors on marine systems. And so we can have all of these types of events that will cause freshening, salinization of different areas across the globe of marine systems. Ocean acidification is obviously a problem that we're concerned with. Eutrophication, global warming, all of these things can influence the physical and chemical properties of the ocean, as well as obviously influencing all of the biological components. When we're thinking about it in terms of the data that we're presenting, this is kind of what I want to suggest. We have these anthropogenic stressors that can influence the system, which ultimately can influence the phytoplankton community composition and activity. So if we have a system that's dominated by cyanobacteria, does that system shift to ultimately lead to a diatom dominated system, which ultimately can lead to differences in overall activity, leading to differences in dissolved organic matter production, lysate generation, the killing of cells, the release of that lysate material, and just overall exudation as those cells are growing. As hopefully I've pointed out, the microbes that are using this organic matter, this DOM, are very important. And the different microbes that are using the different sources of organic matter can have important implications on how that organic matter is used and ultimately determine how that carbon flows through the microbial loop into the larger aquatic food webs through trophic transfer events, through moving that carbon into the biomass, or through remineralization and respiration. Ultimately, all of these differences, all of these shifts in how that carbon is transferred can, again, impact these overall physical and chemical processes and lead to cyclic feedbacks that drive system perturbations and ultimately influence the overall functioning in global carbon cycles. So that's where I want to leave it. I'll just finish with some acknowledgements. Key acknowledgements: Brandon Keefe, Sam Bryson, my past students, and Joe Lee, who was doing all the proteomics work at Oak Ridge when he was a postdoc there. Joe Vanoni and Halsey Labs for helping us with some of the equipment and strains that we used. We collected and set up some of these experiments at Hatfield, so thank you very much for that. did a lot of sequencing at CGRB, and funding mainly from the Gordon and Betty Moore Foundation for this work. So 5 minutes left. I'm happy to take any questions that you have. Thank you for your patience and attention during the talk.
Thank you very much. We are a group that I think I can go ahead and allow you to unmute yourself. So I am going to activate that. So if you have any questions, you should be able to unmute yourself. If you'd like to ask them over your mic, you're also welcome to put them into the chat box if that is a more comfortable way for you to interact with Ryan. Ryan, the other question I was going to ask, if folks did want to follow up with you, did you have contact information you wanted to share? I didn't see it on your last slide. I might have missed it, but you could also put it in the chat.
Yep, that's fine.
Great. And let's see, got anybody? I don't want to miss somebody as I'm scrolling through. So this is kind of a different language for me, so forgive me if I've missed something as I'm processing. But I am curious, the work that you've done is primarily at Equina, and you've alluded to the fact that the functional groups that you see in Equina, you expect to see in other places, because that function is what is needed as a community-wide. But I'm curious about next steps. Are you thinking about validating that assumption and doing sampling in other places, or kind of what is the next step with your process?
So there's certainly a lot of sampling that's been done by us in other locations in the Pacific, but also by many other groups. There's some wonderful global sampling projects that are meant to try to ask the question of microbial community diversity across ocean scales. And so what we see here at a very high taxonomic level, of course, is that our samples are very similar to many other samples. You're obviously going to have big differences or bigger differences occurring when you get from the coastal to the open ocean that can drive things. But at least the main point that I wanted to get at here in this talk is that our samples weren't very different. And so the things that we're seeing here likely will apply to many other organisms. And one thing that I didn't touch on here is something that was explored in a previous study of ours, where we were looking at essentially conserved patterns of resource use across different taxa that are phylogenetically related. And so the idea here would be, if you have a known set of taxa in Equina and you have relatives to that taxa in Monterey Bay, are they doing the same thing? And basically what we've seen so far is that there is high conservation of the assimilation that we see, meaning that if we have similar taxa in point A versus point B, we can make inferences about point B from our experiments in point A.
Nice. So we have a question in the chat box, if you can see that, from Fiona.
Can you give us some global numbers of percent of contribution of different types of phytoplankton on global carbon cycling scales? I don't have that information off the top of my head, so I'd have to look that up, but that's a good question. It's definitely something to be aware of, and thinking about how those proportions are going to change relative to changing environmental conditions. And that is really kind of the next step in my thinking, in considering how we might manipulate the system through experimental manipulations and track carbon flow as a response to shifting phytoplankton community composition, which affects the microbial loop.
Yeah, I was thinking about the different stressors in the response of the community and how that would change depending on, especially even inland bays where the stressors are slightly different and might cause a different response.
Absolutely.
Nice. All right.
Hi, Ryan. This is Jim Caldy. Man, that is really cool.
Okay.
That is really cool. The combination of those techniques and the power to be able to show which groups are actually using what compounds, that's amazing.
I agree.
This is really interesting stuff and just blows me away. You know, and I don't have any real question here. I just wanted to say that this is really pretty darn neat, and I see a lot of power in this tool. going forward. So thank you very much.
Absolutely. And thank you for commenting on that, because I'm always impressed every time I look at this data, how rich it is and how much information. I mean, this is a 15-hour experiment, and we've been working on this data quite literally for years now. So you can wring a lot of information out of this data. And I'll just also point out, this is a marine audience, but we're not just limited to trying to think about this application to marine systems. And we have a grant in review right now to thinking about nutrition in the human gut and mammalian gut and resource partitioning there. And so we really see that as a ripe area to extend this methodology to, but it could really be applied to any system where you have significant amounts of biomass and activity.
I will also just throw out that Ryan is planning to be around Hatfield a little bit more often for the next 2 years. So maybe we'll be able to interact face-to-face a little bit more in the future. And if you have questions, we might be able to just happen to see him in the hallways, which is, what some of the great conversations are happening there.
Yeah, that would be great. Yeah, I was hoping that I gave this talk about a month ago, and I was hoping to get together a talk on seagrasses, which is involving work with Fiona Tomas-Nash and myself. And that's the work that we'll be continuing. So hopefully I'll be able to come back and talk to you all about that work one day. Seagrass microbiomes.
Always welcome. It'd be nice to hear follow up on some of the work that Winnie was doing, as well as just how that's progressing and adding to that information.
Absolutely.
Wonderful. All right, everyone. I'm not seeing any other questions. I'm going to give an awkward pause to see if anybody unmutes. All right, we'll call that good for today. Thank you everyone for joining us. Hopefully you'll be able to join us next week. And again, Ryan did put his information into the chat if you needed it. And Ryan, as you see, you're starting to see thank yous come in. So everybody's doing their virtual clap. So thank you very much. I appreciate it. Everybody online, have a good rest of your day, and we'll see you next week. Thanks, Ryan.
Bye.